Hi, my name is Brian Fife. I'm an assistant professor in the Department of Medicine here at the University of Minnesota. This project specifically will look at the HLA genes, those genes associated most highly with type 1 diabetes, in a mouse model setting, so that we can look at the proteins and targets of the immune system in this mouse and ask these questions without any risk to the patient. So if we're successful, we'll be able to shut down the immune system, a human immune system that can cause diabetes in a mouse. The partnership is I think, a perfect one with Mayo. They're experts in genetics, in human genetics, and they've created throughout the years models of studying diabetes using human genes. And we're experts in cellular immunology, so we understand very well how to activate the immune system and more importantly, how to turn it off, so inducing immune-specific tolerance. And so this is a partnership that takes both our strengths and combines them, I think, in a very nice system where we'll look at the immune targets and shutting off autoreactive type 1 diabetic causing T cells in a system where we have the human components of type 1 diabetes. And with this grant, we'll be able to move forward very quickly with human relevant studies, something that I truly believe will be translational in nature and have an impact on type 1 diabetic patients.
We will be mainly addressing mechanisms to induce tolerance to human T cells, so that we can prevent diabetes in the patients. So that is the ultimate goal. So for this, we will be making use of what are called the humanized mice. So these humanized mice express the HLA molecules, which are more commonly seen in patients with type 1 diabetes. And these HLA transgenic mice are also immunodeficient or immunoincompetent. So we can transfer human T cells as well as human islets into these mice. And we propose to induce tolerance in these T lymphocytes to various islet associated antigens. What happens in type 1 diabetes is that our own body's defense mechanisms, they start attacking the beta cells of islets of Langerhans, which are specialized cells which produce insulin. Now, when these beta cells are destroyed, our body is no longer able to produce insulin, which is a very important hormone. So our body is not able to maintain an adequate levels of blood glucose in the system. So the patient invariably needs exogenous source of insulin lifelong. We can transfer human cells and tissues into these mice. So what we plan to do is we transfer human T cells isolated from patients with type 1 diabetes into these mice. A normal mouse would kill these human T cells because it is a xenograft. So now, because these HLA transgenic mice are immunodeficient, they can accept human grafts. At the same time, we will also transfer human islets into these mice. So a normal process of similar to autoimmune type 1 diabetes would occur in these mice. So the human T cells would attack the human islets, which expresses the HLA molecules. The same autoimmune disease occurs, but now it occurs in mice. Now we can test our various protocols which we have designed or developed to induce tolerance in those T lymphocytes. So now, if our strategy works, then we will have different groups of mice. One group of mice will undergo our scheduled treatment, and those T cells will be tolerized. And those T cells will be unable to reject the islet or mount an autoimmune response against the islet. So they will not get any diabetes. We bring investigators from Mayo and investigators from University of Minnesota. We bring in the different resources. So this is an excellent way to do research, bring in different brains together and different technology. So we provide the technology, what they don't have, they provide the technology, what we lack here. So together we hope to have a full functional research team, so that we can kind of tackle this problem and get it to human use. Our long-term goal is to develop this so that it can be used safely and effectively in humans, either to prevent and to treat type 1 diabetes.